the fundamental thing we are chasing in the lab is we think that one can learn a lot about, fundamental biology from a disease perspective. Dysfunction teaches you a lot about function. So the way we are trying to understand is we're trying to model or disease model the disorder in a dish or from a animal model system that not just gives us insight into what could go wrong in a disorder, but it is also revealing very novel insights into how human brain development happens and all of that.
Good morning, everyone. This is Sugandha, Asthana, your host for today's PhD talk show hosted by Biopatrika. Today we have with us an eminent guest, Dr. Bhavana Muralidharan. She is currently working as an faculty at InStem. So welcome, Dr. Muralidharan, and thank you for speaking with us today.
Thank you so much for the invitation. It's an honor and privilege to be talking to you.
Thank you. So, Dr. Muralidharan, I just wanted to start off the interview by asking you, how was your journey from your PhD to your current position at InStem?
Okay. It's been a journey of slight twists and turns in the sense that I switched. So my PhD was in a very hardcore molecular biology question, where I'd studied RNA protein interactions. Particularly, I was looking at how the translation regulation of insulin mRNA happens in response to glucose in the pancreatic islet cells. And it was a very molecular type of study where I had characterized the secondary structure of mRNA, RNA binding proteins, interacting with the mRNA, upregulating the translation, and like that. Towards the end of my PhD, I had already started thinking of my work, wanting my work to have more specialized, or I would say perhaps something which can go into an application-based or a clinical side to it. So I felt studying developmental neurobiology, particularly how brain development happens, is a very good starting point because, with that kind of a knowledge base, one can think of applying things to studying neurological disorders and so on and so forth. But clearly my training in my PhD was very, very helpful because for many reasons that, a direct advantage was the fact that since I was a trained molecular biologist, I could ask molecular questions in neuroscience. And that was the immediate first sort of advantage of having been trained as a molecular biologist. But clearly, PhD equips you with a lot of skills, creative thinking, out-of-the-box thinking, being at a problem, tenacity, persistence, so many things. So all that also really helps when you actually start a postdoc. And my postdoc was with Professor Shubha Tole at TFR Mumbai, where I was studying neurodevelopment or brain development from mouse genetic models. And as I was finishing my postdoc, as I said, again, I wanted a disease perspective. So I did a very short postdoc at UKDRI in Adrian Isaac's lab at UCL London, where I'd studied pathological mechanisms of ALS, which is a neurodegenerative disorder. And I'd used iPSC-derived neurons, spinal cord motor neurons, as a model system. So with all this sort of their background, a molecular biology background, a developmental neurobiology background, and a disease background, I could start my own lab here at InStem. My lab now uses all these expertise in asking questions about neurodevelopmental disorders. Particularly, we're interested in intellectual disability and bipolar disorder. So we use both mouse model system, and we also use iPSC-derived organoids and 2D cultures as a model system. So that's the sum total of my journey and how I now come here to InStem as a PI.
Oh, that's really great to hear. It was a long, I think, but it was a fruitful journey. And how was it that you chose your career in academia?
I wouldn't say it was not something I wanted to do in school. Clearly, in school, I was very as like most most students, I was wanting to become a doctor. I given pre-medical tests, the pre-medical entrance exams also. But since I didn't get it in that particular year. But got, I got the admission to a BTech biotechnology program. And in that program, in general, I was very much interested in science, I would say, even in school. Physics, chemistry, or biology were some of the subjects I was very much interested and excited to learn more about. So the seeds were always there that, science was like something which I had grown to understand and appreciate and be excited about. So in the BTech program, we had very nice subjects on molecular biology, biochemistry, genetics, and developmental biology, where we were taught how all of this which we are seeing in the textbook came from research. So that sort of planted the idea that, this is something I can also think of, is something I can explore and see whether is research something I would like to do in my as a career. So towards the end of the BTech, I was almost clear that I do want to do a PhD. So that I can see whether is research and career option or not. So that's how I opted for a PhD. And then in PhD, of course, the interest was really kindled, and then I decided, okay, why not try to be an independent PI and see how one step led to the other. It was never a conscious decision that I only want to become a PI, so this is what I'm going to do. But one step after PhD, then postdoc, and then second postdoc, and then the PI thing happened.
And what scientific discovery are you chasing right now in your lab?
So brain is sort of like a black box, because access to the brain and studying the brain is really, really difficult from a human perspective. So we have mouse models where one can look at it, but clearly recent studies show that the mouse and the human brain, particularly during development, are very, very different. Right. So then iPSC-derived model system like cerebral organoids or 2D neuron cultures are a very good model system to study these. So the fundamental thing we are chasing in the lab is we think that one can learn a lot about, fundamental biology from a disease perspective. Dysfunction teaches you a lot about function. So the way we are trying to understand is we're trying to model or disease model the disorder in a dish or from a animal model system that not just gives us insight into what could go wrong in a disorder, but it is also revealing very novel insights into how human brain development happens and all of that. So to put it in a nutshell, we are trying to human in vitro disease model of neurodevelopmental disorders. That's the sort of big project in the lab. But on the way, we are also uncovering fundamental mechanisms in human brain development, which we are trying to get it from, a disease perspective.
Yes, okay. That's great. So how is your journey as a PI? What does it feel like mentoring students and, just guiding them along their journey of PhD?
Yeah, I would say it's a pleasure because to be interacting with young minds is always a source of great joy. But also come along with it, it's a responsible job as well. In the sense that right now I'm at the bench teaching them how to do experiments. You have to, they are very curious, they're very, enthusiastic, these young minds. So and it's also your duty to shape it well at this juncture, which means that, not just telling them how to do the experiment, but also to ignite the spark that how to think about the research problem itself. Because you in the school or even in the college, you're taught in a certain way. And sometimes that way is typically rote learning and not creative thinking or thinking about a hypothesis driven thinking. So one needs to be sort of inculcating right from those things, right? How do you start thinking about a research problem? How do you work at the bench? What are some of the good lab practices? Some good lab practices also involve writing good lab notebooks and all of that. So one needs to, sort of teach them all this. But I think also very important to tell them is, and this is something I keep telling to all young people who work with me, is that, research is a marathon. It is not a sprint. So never think it's a sprint. You need to pace yourself very, very well, so that, it's one step at a time, so that, you one step at a time, so that you daily build on the research. You don't suddenly reach the research once you did it something today, tomorrow you don't have a result, right? So that attitude is sometimes very important to sort of tell them and keep telling them again and again, because often we see failures and then some success. So one needs to be prepared for that as well. So these are some of the things which I find, as a responsible job which I need to do. But of course, it's a pleasure working with all of them. They come up with such great ideas and so much enthusiasm that, you feel great to be working along with them.
Great, great. And see, I'm at that juncture right now because I'm just finishing my PhD. And if what advice would you have for people or students like me who are trying to move into the academia after their PhD?
Yeah. So I think choosing the postdoc is a very critical, I would say, a critical decision. Because in some sense, it also determines what you're going to be working at least in the next 5 to 10 years. PhD is more like a learning time point, where if you also, if in that time period also, this is what you want, this is the area you want to be working on, then it's fantastic. Otherwise, this is also a time point where if you want to switch, this is a good, time to switch when into a new field or a new area in the postdoc. Having said that, it's also important, you need to be choosing your labs very carefully. If you want to stay in the academia, you definitely want to do a good postdoc in a good lab, where you need to be choosing a good mentor, a good lab which is funded well, which has good research questions, where your career growth is that you will have a career growth in terms of getting independent projects, in terms of getting the funds to do the project, in terms of thinking about good publications. So I think it's very important to spend some time choosing a good postdoc lab. And I would say it takes about a year to come to that decision. So never do it in a rush. Always in the last year of your PhD, start thinking on where do you want to switch? Do you want to switch the field? If you want to switch the field, then use the last year as a networking time where you reach out to people in the field whom you want to talk. Need not necessarily be a PI, it can also be another postdoc, or even a graduate student from the lab where you want to discuss and understand what is the kind of science being done in their lab. And potentially everybody likes a year's advance time to decide on a postdoc, good postdoc candidate. So it's all, time is very important. You need to be starting everything in advance so that, you make a good choice and you don't make a choice in a rush. So I would say start a year in advance, start talking to people, start networking, start reading other labs' work. You come up with a small SOP which you can share it with your potential prospective PI. They also like people doing their homework. So all this is important.
Okay. See, nowadays I've seen a lot of people are moving from academia into the industry. But is there any way to move into the academia after you have like spent some time or experience in an industrial background? Can that be done?
Um, I mean, I think it should be doable. Absolutely. See, I'll tell you why I think that. Because PhD is not just a degree, right? It's a life skill. It's a skill which is never going to go away from you, no matter whether you go into the industry or not, right? It's a skill. See, if you've spent 5 years working on a research question, it definitely tells you have the patience to do it. Which also tells you that you've seen many ups and downs, and in spite of all of that, you've been successful in doing that. right? It also tells you that you're productive because you've been at the bench working, doing experiments, and not just experiments, you've also been thinking. So you also have the creative side to it. So all of this is not going to be wiped out with a stint in the industry. So academia or non-academia, I think wise people everywhere understand that PhD degree is a life skill, and people who successfully done a PhD know what they're talking about. So I think it should definitely be doable. So, I mean, I don't think there should be any, restrictions on, if you've gone to the industry, you can never come back to the academy. If at all, it should really help you in the academia because you would have picked up something from the industry, and that will definitely be useful in the academia.
Great. Okay. So everybody talks about work-life balance nowadays, work sometime, enjoy your life the other time. Do you have any other hobbies that you like to follow besides working in the lab?
Yes, I used to be a trained class. I was a trained classical dancer, and yeah, and for a long time I was in practice, but not anymore. But these days, my hobbies include running, playing badminton. So these are some of the things which I pursue in the side. And you rightly said you need to unwind, where you're completely off from lab and research, so that you unwind completely, and when you come back, you're coming back charged. So it is very important. And so I spend my weekend, Sunday, actually playing badminton and running. So those are the kind of hobbies I have.
Great. So I think that's it for me. Thank you, Dr. Muralidharan, for speaking with us today. It was really an enlightening talk. It was like a window into the academics, and I think it would help a lot of students who want to pursue their career into academia. Thank you once again.
Thank you so much. Yeah, thanks.